Aitwa Ben nani?
How do you work?
Kuna haja kusumbuana na mtu. Si ndio?
I don't know why my slide is not moving. Let me... Oh, just moved. I don't know. I hope the internet is okay. I mean, you can... Genes actually interact. Then under this, we're going to talk about one form of gene interaction is actually epistasis. Then the other one is pleiotrophy, polygenic traits. Those are the three main. There are so many gene interactions, but we are going to focus on maybe these three. And we also look at factors which affect Mendel phenotypic ratios, because some of the gene interactions are the one which affect Mendel phenotypic ratios. You know, the phenotypic ratios, for example, when you are considering a monohybrid cross, whereby you are factoring only one trait, for example, flower color or something like that, you expect a ratio of 3:1 in the F2 generation. Or when you are considering a dihybrid cross, whereby you are factoring two traits, you expect a ratio of 9:3:1 in the F2 generation. We are saying that there are factors which alter Mendel's phenotypic ratio. So those ratios can be altered by gene interactions. So you are going to find that these gene interaction, epistasis, pleiotrophy, polygenic tree, they actually affect the Mendel's phenotypic ratios. So we start right away. So sometimes the expression of one gene can affect the expression of another gene in an organism. You see, from the one interaction, we mean two genes which are actually interacting with each other. So you find that gene A is interacting with gene B. In other words, you find that when A is there, G can be suppressed. Or when A is not there, gene B cannot be expressed. So there is a form of interaction. So this interaction you cannot predict by Mendel phenotypic ratios laws, because Mendel, they only focus on the trait, and they look at the trait, how it appears in the next generation and maybe the next generation after that. But if you have some genes masking or affecting those genes, you find that the Mendel's laws will be affected, because maybe a particular gene may not be expressed in the new generation. Then you find that the ratio will be affected. So we have epistasis, polygenic traits, and pleiotrophy. So we are going to focus on these three. So we start with the epistasis. So epistasis is actually the interaction of two or more genes to control a single phenotype. Two or more genes interacting to control a single phenotype. Maybe we are talking about an example we can talk about. This is an example, it's not in humans, but for example, this is an example in coat color in horses. You find that is like two genes. Actually, you have the gene at locus DUC, BBC. So two genes. This is one locus. You can see the two alleles, BB, then CC. So this is one locus, another location. So they actually interact to control the coat color of the horse. So usually a gene at one location or locus affects the expression of another gene at a second locus. So when you have that kind of interaction whereby one gene is affecting the other gene in a different locus, we call that kind of interaction epistasis. For example, coat color in mammals, and specifically maybe horses. So in horses, you can see from the right, you can see we have horses of different coat colors. We have the black, or we have the brown, then we also have the tan, we also have the white horses, the white horses. So brown color, brown allele B is actually dominant to the tan. You know what I mean by the tan color. So when the horse has the B allele, like in this case, this horse is likely to be what? Brown. Then when he has the recessive allele, that is the small b, the horse is likely to be what? Tan in color. But the dominant, we have now, that is one location. We have another gene at another locus, or we have alleles at another locus, now C. So we have a dominant C allows pigment to be deposited. That is now this pigment, this brown pigment, not the tan pigment. its deposition will depend on whether the host has the dominant C gene on that other location. So dominant C gene allows the pigment to be deposited, while the recessive will prevent the pigment from being deposited. So you can see these two genes in the two different locus are actually interacting. So it means what? When the host has the recessive C, no pigment will be deposited, whether he has the capital B for brown color or the B small b for tan, you find that the color will not be deposited. So the brown color and tan color will only appear when the host has dominant C on the other locus. So homozygous recessive, that is now the CC, leads to an incidence of white horses, because you see the recessive gene doesn't allow any color to be deposited. So in such a scenario, you find that, for example, this form of a dihybrid cross, whereby we are crossing these two parents with this form of allele combination, you find that those organisms with the recessive C will actually be white. You can see here, where the other locus has the recessive C, both alleles are actually recessive. CC, all these horses will actually be white because CC prevents pigment deposition. But when we have a capital C, okay, let's start here. When you have a capital C and a big B, it means now the horse will be brown because B is for brown. But when you have a small b and a capital C, it means the horse will be tan, like here. Capital C, which allows pigment to be deposited. But which pigment? The pigment is specified by the other allele, that is the small b. So B allows for the host to be tan in color. So you can see essentially the phenotypic ratio, assuming this is the F2 generation. So the phenotypic ratio has changed from 9:3:3:1 to 9:4:3. So epistasis is a form of gene interaction which will actually affect the Mendel phenotypic ratios. I hope you have understood that. So as long as you have a capital B, it means the house is likely to be black. But that brown. But that brown color will depend on whether on the other location we have a capital C. So if you don't have a capital C, like in this case, it still has the B gene for brown color, but there's no capital C, so the horse will never be brown. It will just be white, because the recessive C will prevent deposition of pigment. I hope that one is clear. We can do more examples. So we have another question here. In dogs, I'm sure you have seen dogs of various colors. We normally call them, they are normally called the chocolate dogs or the chocolate labs. Like this one, we have the dark ones, we have the white ones. Then this is the common ones, which you normally see. So coat color in dogs is also determined by two genes. I'm just giving you examples of maybe animals, so that you can actually understand everything. Because I'm also going to give you examples of the epistasis actually in humans. So we start with some of these easy to understand. So in these dogs, they are known as the retrievers. In dogs, coat color is actually controlled by two alleles. We have a dominant allele E, which produces a dark pigment. Dominant. So when you have an E, the dog is likely to be dark. While the recessive E produces little or no pigment. So the white dogs, you find that allele for coat color is actually a recessive gene, which is now we are giving it as the small e. So E will prove. In other words, the dog will look somehow whitish. But when the E is there in dominant, the dog is likely to be what? Dark in color. Now we have a gene at another locus known as allele B, so which determines how much the pigment is produced. So when B is there, pigment will be deposited. So when B, for example, when B combines with an E, you see E is for dark pigment, and we have now a dominant B. That dark pigment will be there. But now when E is there, E is for dark pigment, but now you have the recessive B. It means now will not be the dark pigment will not be deposited, because now the dog has the recessive gene, which the one which determines how much of the pigment should the dog have. So you find that dog will be actually chocolate. But now when you look on the other side, remember E is for dark pigment, and these dogs, they don't have the capital E, so they have the recessive gene. So they have their coat color is somehow, they have little or somehow whitish, little pigment or somehow whitish. They look like this. But when now they have the other location, they have a capital B. B determines how much. So you find that although it's somehow whitish, when it has B now on the other locus, you find that it will be more white. But now when it has the recessive B, it will still be somewhere there, not that white. So somewhere there. I think you can see how the two genes are actually interacting. I think these are the guys we normally see. Although we have some of these dogs, I think we have seen all these kinds of dogs. So you can be asked, what percentage of puppies or dogs will be chocolate in color if you crossed two parents that were heterozygous? That is, if you crossed these two parents, these are the hybrid cross, so it will be EEBB times EEBB, the same thing. This is the one parent, then the other parent will still be the same. So because you've been told that gene E is gene B is actually determines it. It is epistatic over E. Or in other words, the two genes are actually interacting, and you know how they interact. You'll be able to estimate the ratios in the offspring. So I think that this question has been done in the next slide. Yeah, so you've been told determine how many dogs will be chocolate in color. And chocolate in color will be where we have the gene for the dark pigment. That is now the capital E, but now the recessive B, which will determine less of the dark pigment being deposited, making the dog to be chocolate. So it will be, you just do the cross. You just do the cross normally. You come up with at least the number of heterozygotes here are 1, 2, the same here, 1, 2. So one parent will have 4 possible gametes, 4 possible different types of gametes, with respect to these locus. So it will be EB, then EB, I think I showed you this last time, then EB, then the other one will be EB. You remember these things. I hope you are practicing, because if you don't practice, so the first gamete will contain EB, capital B, that one. Then the second one will be capital E, small b, this one. Then the third one will be EB. That's how they combine. The law of independent assortment. Then the last one will be EB. So because the two parents are the same, you find that the female parent will also contain the same kind of gametes, will also produce the same kind of gametes. So you place one parent here, the other parent there, then you just push them. But because you've been told that there is some form of gene interaction, at least this one will be dark in color, because B will allow the dark pigment to be deposited. So as long as you have a capital E and B, that dog will be black in color. But as long as you have a capital B and a small b, that means now the dark color will be deposited, a little amount of the dark color will be there, so that dog will be chocolate. So at least when you have a EB chocolate, this one will be dark. Then this one is now this one is white, but this one is now the extremely white. Somebody is watching movies. So this one is the one will be the blue one, because I cannot display the white. So the blue one will be the extreme white. Remember, B determines how much of the pigment is deposited, and E is for little or no color. So this will be extremely white. Then this one I think will be the other one, the brownish, this one. So I hope you have understood. So epistasis is interaction between two genes to control a particular trait. So we can now define some terms. So we can define what is epistatic gene, what is hypostatic gene. So the alleles that are masking the effect are called epistatic genes or alleles. Like now we have seen B is actually masking the effect of the coat color. the effect of E. So we can say B is actually epistatic over E. So the alleles that are masking the effect are known as the epistatic genes. Then the alleles whose effect is being masked are called hypo. Like now E, the coat color will either be masked or will depend on whether B is there. So the alleles whose effect is being masked are called hypostatic genes. Then you can have dominant epistasis, whereby the epistatic gene will exert its effect, is actually its gene or its effect with the presence of a dominant allele. Like in the case we've been shown you, we have seen that it's either when we have the dominant B, the coat color will be deposit, or when we have the dominant C, maybe the horse will be what? Brown. So in dominant state, so when the epistatic gene exerts its effect with the presence of a dominant allele, we say that kind of epistasis is known as dominant epistasis. But on the other side, when we have the recessive epistasis, whereby the epistatic gene will exert its gene with a homozygous recessive genotype. I'll show an example of a recessive epistasis, the way I've shown you. The two examples I've given above are examples of dominant epistasis. But in recessive, you find that the epistatic gene will mask when it is actually in its recessive form. So, for example, in dominant epistasis, you find that A masking the effect of B. You see both of them. A is actually dominant, it's also masking a gene at another location. The other one doesn't matter. As long as you only look at one gene, the gene which is actually masking should be in dominant state. So A dominant masking the effect of another gene at another location, maybe gene B. Then a recessive epistasis, whereby which is caused by a recessive allele. So the recessive allele, they have to be there in recessive form so that they can actually mask the effect of another gene in another locus. I hope you are following. Therefore, under epistasis, you find that Mendel laws about one gene are changed by the second gene. So you find that one gene will be affected by the other gene. So for example, gene C controls the color of person's eyes. Yeah, you have the blue eyes, you have the brown eyes. However, gene A causes albinism. So you know the albino gene. People with the albinism, they actually, you find that that albinism gene is actually epistatic over very many genes. So you find that the high color will still be masked, and you find that the high color will never show any pigment. They'll actually appear some orange. They'll take the color of the blood or something like that. So albinism gene marks many pigmentation genes in the body. So you can say the albinism gene is actually epistatic over, it controls other genes in the body. And it's actually in a recessive state. Yeah, for somebody to have albino, I think he has to carry forward it should be a small AA in a recessive state. In other words, this is an example of a recessive epistasis. So it actually should be a small a, but anyway, this was just an example for explaining further epistasis. So you find that small will mask the effect of very many other genes in the body, and the person will never have melanin, even the color will not, the eye will never have any eye color, etc. So therefore, if a person is carrying gene A, it will no longer matter which genotype or gene C is carrying, and the eyes will have no color, they will just be red. Then epistasis involves one gene affecting, this is now another or just a further definition of epistasis. Epistasis involves one gene affecting or masking another gene, or two genes controlling the same phenotype. So you can use the first definition, one gene affecting or masking another gene, or you can say two genes which actually act together to control a particular phenotype. In other words, two genes complementing each other. We are going to see an example of a complementary gene action, whereby gene A has to work with gene B to control a particular phenotype, which is also still epistasis because it's a form of interaction between two genes. So Mendel laws are still working for each individual gene, but the phenotype is not determined by that single gene. by that single gene. It has to be determined by the two genes, or the two genes have to interact to determine a particular phenotype. So this is now an example of epistasis in humans. Is actually the interaction between hair color and baldness. Baldness, those people without hair and those people with hair. Some people love red hair. I think this is now with the Europeans or even in Africans. And you have seen these varieties of hairs. So a gene for total baldness would be epistatic to one for blonde hair or red hair. So the gene for baldness is usually dominant. It's actually, it shows some form of dominant. We can say dominant. It exhibits dominant epistasis over the gene for hair color. So hair color genes are hypostatic. You see, they are the one which the effects are masked by the baldness gene, which is actually the one of the epistatic. So the hair color genes are hypo, the baldness gene are epistatic. So the baldness phenotype supersedes, or it actually replaces the gene for hair color, and so the effects are not additive. So you cannot have baldness and maybe hair or something like that, hair color or something like that. In other words, one gene has to replace the other one. One gene has to replace the other. So you find that when you have this gene for baldness, you find that sometimes it may not be expressed when you are very young. So maybe later, when it is actually expressed, you find that maybe the hair will actually disappear or something. I don't know whether I'm confusing. We're talking about baldness and hair color. Anyway, the thing is, just know that the two genes are actually the hair color. Actually, this one is the baldness gene is the epistatic. The hair color is hypostatic. So once you have this gene, you'll never have any hair. So if you have the gene for baldness, you will not have any hair. Once it is expressed, your hair color will disappear, whether you had blonde hair or red hair. So this gene is dominant, epistatic over the gene for hair. You can just say the gene for hair, because whether you have red hair or blonde hair, or when the gene for baldness is actually expressed. So these are two genes which are actually interacting with each other to control whether you have hair, red hair. So, okay, epistasis is also, this is another example. It's also seen in people with red hair. Okay, the individuals are homozygous for the red hair alleles. Now, this is another example, different. So we are just focusing on now the hair color only here. So it's seen in people with red hair. The individuals are homozygous for the red hair alleles. So we are saying that is for you to have red hair, you have to have both the recessive, the alleles for red hair be in their recessive state, and they have to be in their homozygous recessive state. So, and when they're in that state, they actually mask the expression of the brown color. So we have another color known as brown color or hair color, which is brown. So it is actually masked by the, when you have the recessive homozygous alleles of the red hair, you find that you will not express the brown color. So definitely you'll have red hair. So at least two genes, this is the point now, at least two genes are involved in hair color. So we have the TRY gene and MCR1 gene. So some people who lack black or brown hair, some people who has black or brown hair is making, or it means that those people are making large amounts of a dark pigment known as eumelamine. So this is now the pigment is there. So if you have black or brown hair, you are producing this pigment known as eumelamine. And we are saying that this eumelamine is actually affected by two genes, TRY and MCR1. So you see, two genes are interacting to determine your brown or black hair color. So to make eumelamine, the amino acid tyrosine has to go through multiple transformations, which involves the use of protein MCR1. It's actually MCR1 gene. This gene produces a protein of the same name which helps to direct the chemical reaction. So when MCR1 protein is made and active, it it pushes the chemical reaction to produce the brown pigment eumelamine. So we are saying that, yes, Kwena Kizito, okay, you have read that. Let me just explain before I give you a chance to ask the question. So we are saying that people with brown hair, they produce what? Eumelanine. And two genes are actually involved, TRY and MCR1. So I think the first step is production of tyrosine, and this amino acid from this gene, TRY. Then that tyrosine has to undergo very multiple transformation. Then before it is actually mixed, now before the tyrosine is converted to eumelamine, there must be this gene which is known as MCR1 gene, which is actually expressed, so that now the tyrosine can later be converted to the black pigment, that is eumelamine, so that you find that you have brown hair. But if you don't have brown hair, it means there's a mutation somewhere, and it actually occurs in the mutation there's a mutation in the MCR gene, so that you don't produce this gene. And that's why we are saying people who are homozygous recessive, you find that they are homozygous recessive, they have an allele which is actually in a recessive state here, so they don't produce what? MCR1 gene. So that you find that the tyrosine is never converted to eumelamine. Instead, it is actually converted to another pigment known as pheomelamine. So when it's not being met, that is now the MCR1 gene is not being met or is not working properly due to change in the DNA. Now maybe those individuals who are possessing now the recessive allele at this state, at the MCR1 locus, so you find that they are not able to transform tyrosine to eumelamine, and you find that now the tyrosine is converted to pheomelanine, and this is now reddish. So variants in X, now those with the mutations here, they don't convert the tyrosine to the eumelamine. Instead, they make pheomelamine. So this is an example of epistasis, whereby two genes are actually interacting to control a phenotype. So when one gene is not there, when MCR1 gene is not there, you find that the individual cannot produce what? Eumelanine, and the individuals will have red hair. So Kizito, you had a question. And if you can see, this is a form of a complementary gene action. You can see this gene is actually indirect. They are actually complementing each other. So whereby two genes complement each other to to express a certain phenotype, we call it a complementary gene action, example of epistasis. So any question, I think now you understand what is epistasis. And the things are not hard. So as Kizito is typing, I think we can still proceed. So this is now an example of recessive epistasis. Remember, I've given you examples of dominant epistasis. Now we need to have an example of recessive epistasis in the blood group. So you know the ABO phenotype, the blood group alleles, the A, B, and O. Then, okay, one of the first epistasis to be described in human phenotype, or the H phenotype. Bombay, you know, Bombay is in India. It was once a slum, and now I think it's somewhat industrialized. They normally call it Mumbai. So the Bombay phenotype, which involving the ABO blood group system, whereby individuals are blood type O. So we are saying people with this particular gene, H phenotype, you see, we have the locus, which controls, we saw that. We have a particular locus in our genome, which actually controls the blood group type. For example, if you have on one chromosome you have A, on the other chromosome you have B, you'll have AB. If you have A, the other one you have O, or you don't have any, you have the recessive gene which produces no antigen, you'll be O. Then B, O, you know, all those things. But we are saying that blood group is not only determined by the blood group alleles. We have another locus known as the H locus in another chromosome. It can be even on a different chromosome, whereby we normally call it the H locus, which actually will mask where the effect of the A, B, and O. So when you have the recessive, let me just read. Individuals with this phenotype lack a protein called H antigen. Now we are saying that in the H phenotype people, or in those group of people, the Mbobe phenotype. So these individuals, they lack protein called the H antigen because their genotype is HH, is actually in a recessive homozygous state. So when that other locus is in a recessive homozygous state, you find that they cannot produce what? H antigen. And this H antigen is the one which is used to form antigen A and B. In other words, it's the one which says, let the other locus be expressed. I mean, let the gene in the other locus be expressed. And in the other locus, the genes were for making blood type A, you have blood type A. Or if they were for B, you have blood type B. Or it was AB, you have blood type AB. But for the individuals who are the recessive homozygous state, that is, they cannot make the H antigen. So you find that whether on the other, it doesn't matter whether on the other locus you have either A, B, or AB. You still be what? Blood type O, because you can't produce the H antigen which controls the production of the antigens on the red blood cell. So will definitely be O blood type. And this one was actually discovered in Mumbai, I think long time ago. So thus, HH is a recessive epistatic, over A and B loci, because antigen will be produced in HH, small HH individuals to trigger the formation of AB antigen. So I hope you understand. So this is an example of a recessive epistatic gene, the HH, because you see it masks the effect of product. I mean, it will mask the production of A and B. When it is in its recessive homozygous state, the individuals, they still be O. They'll never be AB, even if the other locus says this should be AB, or this should be O, or this one should be B. I hope you have understood. So this is the quiz. So ABO, you can be given a question like this. ABO blood group is one of the set of blood group antigen, you know that. And then there is a separate locus H that controls the production of these ABO antigens. So individuals who are homozygous recessive, HH, on that other location, they cannot produce antigens of ABO type, and they always appear O type. So consider the following cross. This is one parent, this is the other parent. So what are the frequencies of their parent blood groups among the offspring? So this one is very simple, because we've been told ABO is okay. There is a separate locus that controls the production of ABO. In other words, there is another locus which is epistatic over the ABO locus. So individuals who are homozygous recessive, they cannot produce what? The H antigen, which is responsible for production of the AB antigen. So such individuals will retain will have the O blood type. So this is a very simple question. So this is one parent, the other parent. Then you do your cross. You form the gametes of one parent, the gametes of the other parent. Then you do the crossing. So from here, which people will be O positive? You can, I'm sure you are listening. You can type in the chat. I mean, not O positive. Just the question is, which people will be O? Will have blood type O? Yeah, from there you can. This is now the total, the possible number of children, or the possible. I mean, the different types of children these parents can actually have with respect to the blood type. So remember, we have said that as long as there is a recessive age, the people will be O. So how many people out of 16? How many children are likely to have blood type O? So am I alone? You can type something. You can talk. Dennis Karanja, David Kamunya. Mulienda. Wekesa. Daniel. Am I talking to anyone? I mean, can you type, say hi, I may be talking alone. Are you there? Hello? Oh, yeah. Please, some people. Oh, okay, you are there, but you cannot. So, how many people will have blood type O? Remember, we have said that when the other locus, that is now the A, is actually in a recessive homozygous state, like in this case you are seeing here, the individual will be O, no matter what. You are saying 4, yeah. No, Vivian, I hope you are giving 4 will have O. Okay, Wango and Kevin, I think you guys are wrong. Yeah, maybe you are just counting this. You are just counting. This one is obvious. Yeah, it's 4, but it's wrong. Yeah, these people, 4, of course, from your prior knowledge, when you have the O, definitely will be O. You'll have the O because you cannot produce the antigen. But now we have an epistatic problem here. I told you whether you are O or not, it will determine whether, we have these individuals, maybe, I don't know, in India or somewhere, whereby they have these locus. I think this locus is still common in all human. We have another locus, HH. When we have a recessive homozygous state, matter what you have in the other locus, the locus for 7. Somebody is saying 7. Yeah, so you have to add the other 3 now. Yeah, this is another. You can see we have the recessive H here, both of them recessive, both of them same recessive. So this guy will not be B, will not have blood group B. This one will not have blood group A. This one will still not have blood group AB. This one will still be O, because this is a form of recessive epistasis, whereby the recessive gene will mask the effect of the expression of the alleles on the other locus. So 7. I hope at least 2 people have understood that. Yeah. So don't say just. But this one, at least you can see a dominant gene H here, dominant H. As long as you have the dominant H, that individual will be able to produce now the H antigen. That H protein, and that H protein will be responsible for, we'll say, let's now make poison on the other locus. If the other locus at A, now this one will be A. This one will be AB. This one will be AB. AB, this one will be B, because H is dominant. This one will be B. As long as you have a single H, then you will get the blood group specified by the alleles on the, by the blood group allele. So they'll actually be 7. Yeah, so 7 out of 16. All those with HH will express the O blood type because they are lacking the dominant epistatic gene. Okay, they are lacking not the dominant. They are lacking the dominant gene, which allows the expression, which allows for production of maybe that H protein, which will now trigger the expression of blood group alleles in the other locus. I think I'll have to change this. So, of course, the OO, this one will be O. This one will be still have the O. Even if they have the H, you see, even if they have the dominant H, and the H protein is produced, the other allele says O blood type. So these are just obvious. This one will be O, but the other guys 3 will still be O. Yeah, so any individual who has OO or recessive HH will produce a blood type O. So I think that one is clear. That's an example of recessive epistasis. So the last one, this one is mice. These experimental animals we like studying. So an example of recessive epistasis is pigmentation in mice. The wild type coat color, which normally known as AGOT, is actually determined by the gene A. Gene A, you can have both A. A is dominant over the solid colored fur. So if a mouse has a dominant A, it will be AGOT. But if he has the recessive gene, both of them recessive, and because A is dominant over A, it will be what? It will be solid colored fur, or he'll have a different color. the fur color or something like that. So, however, a separate gene C, now we have a gene on another locus, is necessary for pigment production. So, a mouse with a recessive C allele as this locus is unable to produce pigment, and the mouse is albino regardless of the allele present in locus A. So this is also another example of recessive epistasis. We are saying that coat colors can either be ergot or solid fur, and we have another locus C, which is necessary. So when the C is there in dominant state, the coat color will be deposited, whether the solid fur or whether the ergot. But when there is a recessive C, you find that this one will mask everything, and the coat color, the mouse will be albino, whether it has the gene for ergot or whether it has the gene for solid fur. So this is also another case of recessive epistasis, because like this individual, you can look, therefore genotypes AACC, ACC, all of them will be of the same phenotype because the recessive C is there. You can see the recessive C. So all these mice will still be albino. But you see, according to here, we are saying that A is dominant over, we are saying that A, the coat color is ergot. Then this one, AA, we are saying the coat color should be solid fur. But because of the other locus, we have the recessive gene. You find that this recessive gene will mask anything to do with coat color, and the mouse will be white albino. So across, you can do this one on your own. Between heterozygotes for both, you can cross this mouse. This is one mouse and another mouse would generate, such a cross would generate an offspring with a phenotypic ratio of 9 ergot, 3 solid color, and 4 albino. In this case, the C is epistatic to gene A. Of course, you have seen that C, the recessive C, is actually epistatic over A. I can see some errors in some of on my notes. I think I'll have to change. At least I can say the recessive gene C, the small c, is epistatic to gene A. So you can do this one on your own. I think you have now understood that. So we are still in epistasis. There's something known as a reciprocal epistasis. Reciprocal, I'm sure you have heard of it in mathematics. So it can be reciprocal such that either gene, one present in dominant or recessive form, expresses the same phenotype. For example, let's look at this example. In the shape of this plant, the characteristic of seed shape is controlled by two genes in a dominant epistatic relationship. When the genes A and B, when the genes A and B are both homozygous recessive, the seeds are ovoid. But if the dominant allele for either of these genes is present, it results to triangular seeds. So this is an example of reciprocal epistasis because we are saying we are using the word either. If the dominant if either of gene A is present or gene B is present, you find that the seeds will actually be triangular. But when both of them are actually lacking, now you are going to get what? Ovoid. So when they are not there, you get ovoid seeds. But when either one of them is there, the seeds will actually be triangular. So this is an example of reciprocal. In other words, A is complementing B. When B is not there, A is doing the work. When A is not there, B is doing the work. So when either of the gene is present, the seeds is actually triangular. But the seeds will only be ovoid when either A and B, the dominant A and B, are actually lacking. You can see that. So that every possible genotype other than AABB now in a recessive state will resort to triangular seeds because A and B they actually complement each other. In other words, they have this kind of relationship. They are reciprocals of each other, if I can put it that way. The B is the reciprocal of A is the reciprocal B. So what would be the phenotype ratio of the offspring for cross between heterozygotes for both genes? So you have been given this kind of the hybrid, two traits. You have these two loci, two parents, focusing on two locus, and the hybrid cross, you do the cross, and then do it on your own, and you are going to find that the phenotypic ratio will be 15 triangular and 1 ovoid. Yeah, I think those examples are more than enough. Yeah, I think we are done with epistasis. Now we can look at the other type of gene interaction. So any question before we proceed to the other kind of gene interaction? So the other kind of gene interaction, we normally call it the polygenic traits. People call it polygenic inheritance. People talk about quantitative inheritance. Other people will also talk about quantitative genetics when they are talking about these polygenic traits. So a polygenic trait is a single trait that is affected by more than one gene. Now we come here to, somebody was asking about skin color, height, eye color. You know, I told you these things like skin color, they are not determined by one trait. They are determined by very many traits. That's why we are using the term polygenes, or we are talking about quantitative inheritance. So very many genes, you can have a gene in chromosome 1, a gene in chromosome 5, 10, 11, 12, all of them determining your height, all of them determining your eye color, or even your skin color. So such kind of traits which are determined by more than one gene, we normally call them polygenic traits. And usually referred to as the inheritance is usually known as quantitative inheritance or polygenic inheritance, which means that the inheritance of traits are determined by more than one gene. These genes are called polygenes. So the genes determining a particular trait, very many genes, if they determine a particular trait, maybe height, we call them, all those genes, we call them polygenes. They produce specific traits when they are expressed together. So very many genes producing height or determining your height, we can call them polygenes. So polygenic inheritance differs from Mendelian inheritance patterns where the traits are determined by a single gene. So you have to know Mendel only focused on one gene. So that's why you find that, these things were discovered later, and they were actually seen to affect Mendel phenotypic ratio. So anything to do with Mendel or Mendelian genetics actually is only focusing on one trait, how that trait moves from one generation to another single trait. That thing about dominance and recessiveness. So Mendel never knew things such as gene interactions. So it's all polygenic inheritance. We are going to see it also alters Mendel phenotypic ratios. So polygenic traits have many possible phenotypes that are determined by interaction among several genes. So we are saying like in eye color, you may find that we have very many genes which are actually eye colors of individuals. So you find that because we have very many interactions, you find that the physical characteristic brown eyes, gray eyes, all that kind of, because we have interaction of this gene. It depends on the genes you get from your parents. If you get more of this or more of that, then your eye color will be this. The same to your skin color. If you get maybe more of the dominant genes, you become black. If you get more of the recessive genes, you find that you are light. Your skin is light. You have a light skin. So I think that one is very clear. So polygenic traits. In these traits, it seems that each dominant alleles has a dose to the trait. Now, that's the thing about polygenic traits. It's like the dominant genes, they had a dose. In other words, like for the case of skin, you find that the more you have dominant genes, the more dark you are. Yeah, you can see, look at the black ones, the black dots. They refer to the dominant gene. These are maybe like a population coming from parents who were actually identical. They had three dominant genes. Remember, skin color is not just determined by three genes. You can see from here, this is one allele, this is one gene, another gene, two alleles of the determine the same trait. Then you have the third gene. You can say these are three genes, the other one. So these are two individuals who are the same. So if you can have such kind of parent, then they make a very large population. It's just, of course, an example. We have, for example, a population of 1,000 people coming from such kind of parents. You find that the more dark you are, like with no respect to skin color, the more dark you are, it means you have more dominant genes for skin color. Then the more light you are, you have the recessive genes. So these are now the light-skinned people, the Europeans, these are now the Africans. But what can you see here? I'm sure you have done some biostatistics. We normally say that normally when the frequency of the dominant allele is actually plotted, you find that you form a bell-shaped or normal curve. You have heard of the normal distribution, the bell-shaped curve. It means what? Here is now the number, the frequency. So low frequency is increased high frequency. So high frequency is around the mean. So a bell-shaped or a normal distribution shows that most people will be somewhere there in the middle. For example, in other words, this means that more people have intermediate phenotype than on either extreme. So very few people, even if you sample the population in the world, and let's say your main objective is to study skin color, you're but we have 7.8 billion people. Very few people have light skin, very few people have very dark skin. So majority of people are in between. They have the intermediate skin color, neither dark, neither too light. So that's the thing about traits. To plot them, you get a bell-shaped curve, whereby most people will display the intermediate phenotype. And you have to know that polygenes are actually additive. That's where dominant. The more you have the dominant, is like you get an extra dose. You have more dominant genes, you have more doses of the vaccine. You have a less dominant genes, or you have the recessive gene, you find that the recessive trait is more highly expressed. So polygenes are actually additive, and their frequency shows a bell-shaped curve. So this is the thing, the skin color. You can see small d. This is now another just example. Small d, small d. The more you have the recessive genes in both chromosomes, the more light skin you are. You see, this guy has one dominant gene, somehow dark. Then two dominant genes darker. Maybe this is now a Chinese or something, or South Korean. Then now we have the more dominant genes. We have something like that. The Chinese, the Indians, and then we are the Africans are here. We have more of the dominant genes. And I told you about this thing. I think it has to do with something like evolution. You see, there's a way when you have a dark skin, there's a way it protects you from sun. Africans, Africa is around the equator, so we have a lot of sun. So I think in the course of evolution, I think this it was more better for survival around the equator. That's why it was retained in the population. And if you have studied evolution, maybe it came from Africa. Then when these guys went to Europe, near the poles, you find that there's very little sun there. So you find that the recessive genes were actually selected because these recessive genes will lead to production of less melamine. And less melamine is needed so that they don't block a lot of maybe radiation from the sun, or they can get as much as they can get from the little sunlight they get, because those people they get a lot of sunlight, unlike in there around the equator, whereby the sunlight is a lot. When these people come to Africa, they get a lot of skin burns, but when you suffer from lack of vitamin D. So the thing is, just stay where you are. So the thing is, there's also another gene for hair color, which is also polygenic. So I think this is also it was another form of selection. There's a reason why these people, they have this kind of maybe for insulation or something, or the same year still insulation, you know, you can read about that. It's also determined by several genes. Yeah, very many genes determine hair color. You see, the thing about genetics is so confusing. The other time we were just talking about red hair and badness. Now here we are saying now hair color is also a polygenic trait. Yeah, hair color is also, you know, of course, you know, we have variation of hair color. Anyway, that other example we were just talking about blonde hair and red hair. But generally, we have a lot of, various types of hair color. We have the, you know, you can get all the varieties. And of course, most people will have the intermediate phenotype, that color which is somewhere in between, not on the extreme, not on the other extreme. We have people with even gray hair. So I think the other one is high color. I think I've explained the two. So high color, you have seen people with all these funny kind of eyes, you know. I think it's very easy to notice high color in the white people because they don't have a lot of melamine like us. But the dominant high color, I think, is the brown eye color. etc. So this is just an example. You can do it on your own with respect to, on high color. So high color is determined by a number of different genes. You see, there are actually 16 in number. 16 genes determining your high color. For this example, we'll assume that it's actually determined by 2 genes. In this case, a cross between 2 individuals with light brown eyes. This is now the genes in the 2 loci, light brown eyes. You cross these 2 individuals, BBGG times BBGG, the same way, will produce several different phenotype possibilities. In this example, the allele for black color is dominant to the recessive blue color. So blue color is actually recessive. So those people with the blue color, it means they have all the recessive genes. They have the recessive gene in homozygous state. For gene A, for gene 2, now gene 2, the one which has GG, the dark hue, G is dominant and produces a green color. Then the light hue, small g, is recessive and produces light color. So the cross will result to 5 basic phenotypes and 9 genotypes. So these people will have black eyes, dark brown eyes, that is now this one and this one, light brown eyes, green eyes. Yeah, these ones are real, but we don't have people with green skin, like I was asking last time. At least we have green eyes and blue eyes. So you can do the cross. You can cross these 2 individuals. That is you can cross the 2 individuals. That is now capital B. The way you are seeing it, B, then capital G, small g. You cross with the same thing, capital B, small b. You know, the same thing. You cross this times another, which is looking like that. So I think we are done with the polygenic. I want to see what is polygenic traits. Yeah, the same thing. This is now height. Height is also another form of polygenic. We have given an example of skin color. The same to height. If you take a large population, a population of individuals, then you measure their height and you plot the number of individuals, the frequency with a particular height. You know, height ranges from, is it around the normal height? Around 5 to 6 feet. So if you plot such kind of that data on height from the population, you're going to get the bell-shaped curve. Very few people will be extremely tall. And you know, those are around 6 feet. Then very few people will be around 5 feet. Most people will be around maybe 5.5 to I think 5.5 to maybe 5 something. They're around 170 centimeters. So that's the normal height. So many people will be there. So now we are done with polygenic traits. Now we have another kind of gene interaction known as pleiotropy. So pleiotropy results when a single gene affects more than one trait. This is actually the opposite of polygenic trait. In polygenic trait, we say that very many genes are single trait. But in pleiotropy, a single gene affects more than one trait. For example, you can have a gene A affecting let me just give you affecting maybe high color. The same gene is also affecting your height. The same gene is also affecting your hair color. So when a single gene affects more than one trait, that kind of interaction, we call it pleiotropy. An example is this one, sickle cell disease. So we'll still come to sickle cell disease. The sickle-shaped red blood, you see, when you have that gene for that disease, the main symptom we know is that the red blood cell will have what? Sickle shape. Will be sickle shaped. Then that gene, apart from forming the sickle-shaped red blood cells, you find that you will also have other effects on other in organs. You'll have organ damage, and then you will also be able to resist to malaria because of the shape. So you can see, these are like from one gene, you are getting 3 traits. Resistant to malaria is a quality, is a trait. Then you can suffer from organ damage, maybe those white organ damage. Then you'll have now the shapes of the red. So this is a polygenic. All this is pleiotropy, an example of pleiotropy. Then pigmentation alleles in cats. So white coloration in cats, those cats which are white, they have this dominant gene. It can be in the heterozygous form or in the homozygous dominant. You find that the cat will be white, but it will also be deaf. So it's actually one gene which is making the cat to be white and also making the cat deaf. So it causes deafness. So I'm sure you have heard of cats which are deaf or something. Then pigmentation alleles in mice. The YY dominant is a gene, actually when it's in homozygous dominant state, it will actually be lethal. So it is causing death. So you find that these mouse are not even born. They died in utero. Yeah, so making them yellow, so you find only the heterozygotes will survive. The heterozygotes will survive, but the homozygotes, which are also yellow, they die in utero. They don't survive. So pleiotropy occurs when one gene controls more than one pathway or is expressed in more than one part of the body. And I think I've given you an example. For example, one gene makes connective tissue needed for lens of the eye. It can also affect the heart muscles, limbs, etc. Therefore, mutation of this one gene will cause effects in eyesight. It will bring some heart attacks, and also weakening of the muscles and limbs. And you see, that's the thing. This is the thing about the worst thing about pleiotropy. Remember, it's one gene controlling very many traits. So if you have a mutation, in that particular gene, you see, you will suffer from a lot of all those traits which are determined by that particular gene will all be affected. So like you'll have eyesight. This is just an example. You can be prone to heart attacks, or even you can have weak muscles and limbs, just because that gene which affects these 3 traits has been affected. So this is just a chart to show you the relationship. This is the normal case of Mendelian thing, one gene effect. But now this is what polygenic. Very many genes, same trait, like very many genes, height or hair color. The opposite, pleiotropy, one gene, very many traits. And this, I think, this is just a mixture of the two. Then remember, we are looking at factors which affect Mendel phenotypic ratios. We also have something known as environmental influence. So the environment also brings about variation. But these variations are not genetically based. They are actually due to the environment. But if you look at the DNA, you'll find that they are not evident, or you cannot trace them back to the DNA. So we have environmental influence, which environment brings about variation. So environment can affect organism phenotype, and we use this term known as phenoplasticity, which is a variation introduced by the environment. So you hear people talk about phenocopies. Phenocopies, these are actually environmental cost traits that appear to be genetic, inherited, or environmentally caused phenotype similar to inherited, but in a real sense they don't have any genetic basis. It's just basically due to environment. I can give you an example of plants. You can see a plant growing in a desert where there's no food, there's no enough, the soil is not fertile, there's no water. Then the same plant, the same genotype, with everything, with the same genome. Maybe growing in a highland where we have plenty of stuff. You find that this one growing in a highland, it grows very tall, it has very nice flowers, leaves. So, but when you compare with the one growing in the desert, it's actually different. But genetically, the two plants or the two organisms are actually identical. So we normally call them phenocopies. They are genetically identical, but different. And when you look at them morphologically different, because environment has actually acted upon them. So these are some of the examples. I know we are doing human genetics, but you see, we cannot specify all our examples in humans. So, for example, lack of water and sunlight affects flower formation, leaf retention. I think I've given you that. Lack of enough water will affect plant growth. Yeah, so if you provide enough water and nutrients, you are going to see some variation. Yeah, you are going to see variation. Even soil acidity affects the color of androgynia flowers. Temperature affects the color of the fur in Himalayan rabbits and Siamese cats. No temperature. Anything brought about by the environment, whether acidity, nutrients, diet, you even know that. Yeah, you can maybe say, let's say you are twins, then or you have twins, then one twins goes to the gym, the other one doesn't go to the gym. So you find that the one who goes to the gym, you see, we are saying that they are identical twins. The one who goes to the gym will have very nice, bone density, very nice physique, But the one who doesn't go there will be, it'll be different. So those are phenocopies. So the two can be termed as phenocopies. So this is just an example of phenoplasticity in this plant. You see, at one point, the plant has purple flowers. At another point, the plant has blue flowers. They are not different. It's just the same plant. It's only that this change is due to condition of the soil. We have some acidity in the soil. So the reason why this flower changes because we have aluminium ions become, I think aluminium ions in the leaves or something, or even in the soil, become soluble in acidic media. Thus, when pH changes from alkaline to acidic, the aluminium ions are absorbed to the plant petal, thus changing their color from pink to deep blue. I'm sure you know those things. Aluminium sulfate, one is blue, the other one is sulfide, I don't know, pink or something like that. So it's just an environmental effect, whereby we have maybe absorption of aluminium ions making the color change from pink or purple to blue. So basically, these are phenocopies. Yeah, they are genetically identical. So we are looking at the factors affecting Mendel phenotypic ratios, or even remember the lecture. It was on gene interactions, distancic pleiotropy and polygeny. Then we also need to look at factors which alter Mendel phenotypic ratio, because these three interactions they also alter Mendel phenotypic ratio. So we are also looking at other factors such as environmental influence. Now we are looking at penetrance. Yeah, penetrance. So what is penetrance? So sometimes the same genotype will not produce the phenotype in all individuals. You see the same genotype. Let's say you have a specific gene, or you have very many people with a particular gene. You are sure these people have that particular gene, but they may not display the phenotype. Maybe that gene was responsible maybe for a certain character or a certain phenotype. Out of 100 people, which you are sure they have a particular gene, then later they don't show that phenotype resulting from that gene. So that's where now the idea of penetrance comes in. So penetrance refers to the probability of a gene or a trait being expressed. In some cases, despite the presence of dominant allele, a phenotype may not be present. What is polydactyly in humans? Extra fingers, presence of extra fingers in humans. I don't know whether you have understood, but looking at, for example, a population, let's say 100 individuals, you are sure they have a particular gene. So according to Mendel, you expect them to show that particular phenotype. The phenotype resulting from what? From that gene. But they don't, not all of the 100 show. So you may find that maybe 80% show the phenotype. So that probability of a gene or a trait being expressed is what we call penetrance. For example, in humans, we have people with extra toes. Like you may be carrying the gene for extra toes. That is now polydactyly, but you may not display it. Yeah, it's actually there, but people don't actually display it. But I'm sure you're seeing people with extra fingers anyway and extra toes. So penetrance is the percentage of individuals who have a certain genotype and show the expected phenotype. So that is now actually the real the way to calculate it. The percent of the people with the genotype and they actually show it. That is what is now actually the penetrance. For example, Medel's traits show penetrance of 100%, but some traits penetrance is not 100%. So it will be out of the, for example, if 80% show penetrance, 80 out of 100, so the penetrance will be 80%. So the percent of individuals who have a certain genotype can show the phenotype. The others, they have the genotype, but they don't show. So you say the penetrance is maybe 80%, 70%. Although some traits they show 100%, but some traits such as polydactyly, the penetrance is not 100%. Let's assume we all have the gene for extra toes, but maybe 20 of us show the gene, or they have the extra toes. So the penetrance will actually be, let's say the number was 50, it will be maybe 20 out of 50 times 100. I think that one is clear. Oh yeah, we have another example here. For example, let's say everyone in a population W carries the same allele combination for a certain trait, yet only 85% of the population actually shows the phenotype expressed from those alleles combination. So the proportion of genotypes that actually show the expected phenotype is known as the penetrance. Large population studies are actually necessary for measuring penetrance. So you cannot determine penetrance by just maybe sampling maybe 50 individuals, and you have to do, you have to have a large population, maybe more than 1,000 or even several thousands. And the studies of penetrance help to predict how likely is it a trait will be evident in underlying alleles. In general, when we know the genotype is present, but the phenotype is not observable, the trait shows incomplete penetrance. And so when the genotype is present, but the phenotype is not, we say the trait shows incomplete penetrance. Basically, anything that shows less than 100 penetrance is an example of incomplete penetrance. I don't know whether you understand that. Like in the case of in other words, we are saying that some traits, they are not expressed in all the individuals who have the genes for that particular trait. So sometimes you have incomplete penetrance, like in the case of polydactyly. You can say some traits they show incomplete penetrance. In other words, this is the thing we calculate. We actually calculate those traits with incomplete penetrance. We actually determine their penetrance. So decreased penetrance, or low penetrance, or that incomplete penetrance means that some people inherit genotype and yet they do not show the phenotype. So penetrance is calculated. I think you now understand what is penetrance. Individuals who have the genotype and the expected phenotype. You have the genotype, then you show the phenotype. Over there, the total number of who have the genotype. Or the genotype. Or the total number of the individuals under study. Yeah. So another example, penetrance of osteogenesis imperfect. So a specific example of is the human-born disease known as osteogenesis imperfecta, OI. So the majority of people with this disease have a dominant mutation, the two genes that produce mutation. These are the two genes which produce, which are responsible for production of this collagen and this protein, cholia-1 and cholia-2. So if people have mutation, if you have a mutation in either of these two genes, you are likely to suffer from osteogenesis imperfecta. So, and collagen is a tissue that strengthens the bones and muscles and multiple body tissues. So people with OI have weak bones, bluish color in the whites of their eyes, and a variety of afflictions that cause weaknesses in their joints and teeth. However, this disease does not affect anyone who has a cholia-1 and cholia-2 mutation in the same way. So this is an example of incomplete penetrance. You can actually be, maybe this one, the penetrance can actually be estimated because you have the mutation in either cholia-1 and cholia-2. So under normal circumstances, we expect you to express what? Osteogenesis imperfecta, whereby you will have all these kind of symptoms, bluish color in the whites of your eyes, weaknesses of bones, teeth, etc. But not all people will, with this mutation, actually have osteogenesis imperfecta. So that's a very nice example of penetrance, incomplete penetrance. In fact, some people carry the mutation but have no symptoms. They're just normal people. They're very energetic. They're not even weak. Yeah. So thus, families can unknowingly transmit the mutation from one generation to the next through someone who carries the mutation but does not express the disease. So I think that's now the problem with the penetrance. Yeah, you have the disease, you don't show it, then you pass it to your children. Now we have another factor which also affects the Mendel phenotypic ratios, known as expressivity, which is very similar to penetrance. So sometimes the same genotype with degrees of the phenotype in individuals. So expressivity is the degree to which a trait expresses among the individual. Unlike penetrance, the expressivity describes individual variability, not a statistical variability among population. So expressivity is the level of expression. Let's say we are saying now the trait is already expressed, but some people will express more, like in the case of maybe extra fingers. Maybe now this one occurs in maybe cats. Cats may have several extra toes. It may have maybe 5. I think normal is 5. Then they may have 6. Some cats will have 7. Others will have 8. So the level of expression is actually what is known as expressivity. So sometimes the same genotype will produce different degrees of phenotype in individuals. So this is what we call expressivity. It's not don't confuse it with the penetrance. So expressivity is the degree to which a trait expression differs among individuals expressing it. But unlike penetrance, expressivity describes the individual variability, not a statistical. So expressivity, the severity or extent of the phenotype in an individual shows. For example, an example of expressivity is actually hypercholesterolemia. I'm sure you understand these medical terms. I think high levels of cholesterol in the system. So you find that people, they have the gene which actually makes them prone or makes them produce high levels of cholesterol, or maybe they make them produce high levels of cholesterol. But you find that now under expressivity, you find that there is a degree of variation. Some people will have this level, others will have more, others will have very high levels of cholesterol. So that's what we mean by expressivity. Some individuals have extremely high levels from birth, others can control the diet and exercise and even lead normal lives, something like that. So you can see there's some form of variation in the individuals with high levels of cholesterol. So that is expressivity. So, and this is an example of expressivity in cats. I think I've given you the expressivity in cat toes. The presence of extra toe on cat paw is a phenotype that emerges on groups of cats that have been inbred for generation. So polydactyly or extra toes phenotype showed, this is our research which was done, polydactyly phenotype showed good penetrance but variable expression because the gene always causes extra toes on the paws, but the number of extra toes varies widely. You see, number of extra toes vary. A cat can have 5, can have 6, another one can have 7, etc. So through this guy who did the experiment, you find that he found that although the dominant allele underlies the cause of polydactyly, so all those cats with varying number of toes, they have the dominant gene which is responsible for formation of extra toes, that is now polydactyly. The degree of polydactyly depends on the condition of the adjacent layered tissues in the developing limb, that is, the expression of the gene in the tissue surrounding the tissue that will become the toe determines the degree of polydactyly. So you find that the cats, they have the dominant gene for making extra toes, but you find that we also have another tissue there around the toe, which actually, depending on how it develops, it will determine whether another toe will emerge or another toe will not emerge, something like that. So the thing is, expressivity is the variation of expression of the phenotype, or the degree of expression of the phenotype. I'm sure you understand that. So penetrance and expressivity both follow Mendel's laws. Both have something to do with the amount of phenotype present. That one is not informative. So I think this is the last one. I know you guys were tired. The last factor which will affect Mendel phenotypic ratios is actually known as genetic heterogeneity. So genetic heterogeneity is a term which describes different genetic mechanisms that produce the same or similar phenotype. So somewhat similar to what we have done before, but I'm sure by the end of the slide you'll be able to understand. So it can be allelic heterogeneity, which occurs when a similar phenotype is produced by different alleles. I think it's very similar to the polygenic traits. Yeah, the polygenic. Remember we said very many genes, they determine the same phenotype. That is under polygenic trait. But now in genetic heterogeneity, we are looking at, for example, under allelic. This is an example of genetic allelic heterogeneity, which occurs when similar phenotype is produced by different alleles within the same locus. So within the same locus, you see, for example, you can have, in the same locus, you can have a dominant gene on one chromosome, on one chromatid, then on the other side, you have the recessive. So you see, the genes are different, different alleles. We have the dominant and the recessive. But whether one is expressed, in other words, each one will still determine the same phenotype. That is actually allelic heterogeneity, which occurs when similar phenotype is produced by different alleles within the same locus. So it occurs when different variants of a single gene locus cause the same or similar phenotypic expression of one disease or condition. So whether you carry the recessive, then maybe your brother or your sister carries the dominant, you still display the same phenotype, because we're only focusing on that particular locus and the genes which are determined by that particular locus. Whether you have the dominant or the recessive gene, we still display the same phenotype. Then locus heterogeneity, which occurs when similar phenotype is produced by mutations in different loci. So you see, this one is now the one which is similar to polygenic traits. You see, similar phenotype is produced by mutations in different loci. In other words, different loci, they determine the same phenotype. For example, a loci in chromosome 11, another gene in chromosome maybe 16, they all determine the same phenotype. That is locus heterogeneity. But now this is where now the difference. I mean, how does locus heterogeneity differ from polygenic traits? So under genetic heterogeneity, different genotypes are associated with the same phenotype. But here there is no degree of phenotype variation. Remember in polygenic trait we say that we have various kinds of combinations. We have people with intermediate skin color, you have the dark, you have the light, somewhere there. Even within those with the dark, we have variations. So we don't have that kind of variation under genetic heterogeneity. It's actually the phenotype, and there's no variation. It's just the trait only. We don't have degrees of phenotype variation, which is seen in the polygenic trait. So an example of genetic heterogeneity is known as retinitis pigmentosa. So retinitis pigmentosa can be caused by autosomal dominant or recessive. You see the case of allelic. Whether you are this one, autosomal allelic heterogeneity, you can have the dominant gene or you can have the recessive gene. You still suffer from retinitis pigmentosa. Or it can also be caused by an X-linked trait. We are going to come to the sex-linked trait. We normally call them the X-linked because they are carried on the X chromosome, which is actually a sex chromosome. So it can also be caused, this disease can also be caused by what? A gene on the sex chromosome. So still it will give you what? Retinitis pigmentosa. So this is genetic heterogeneity because the same phenotype, retinitis pigmentosa, can be caused by either a dominant or a recessive gene on autosome. You know those chromosomes, number 1 to 22. We normally call them autosomes because they don't determine sex. So it can be, I don't know where it's located. It can give you the disease. And in such a case, if they are on the same locus, we call it allelic heterogeneity. But it can also be caused by another gene in the sex chromosome. So if you can say that is a case of locus heterogeneity. When you compare the autosomal gene and the sex chromosome gene, you can say that's a case of locus heterogeneity because a similar phenotype, retinitis pigmentosa, is produced by mutations in different locus. That results Hello, I think I went off. So this is just a summary of the gene interactions and also the factors affecting Mendel phenotypic ratios. I'm sure you can define them: phenocopies, pleiotrophy, epistasis, all of them here. So next week we are going to talk about the cytoplasmic genetic systems or extrachromosomal genetic system. We are going to see that we have some genes in the cytoplasm. Remember we said that mitochondria as DNA. You have heard of the cytochrome oxidases, the cytochrome enzymes, which are very important in drug metabolism. So they actually produce, those cytochromes are actually, those enzymes, we normally call them cytochromes, cytochrome enzymes are actually produced in the mitochondria. And you know, an enzyme is a protein. And later we are going to see the process of protein synthesis, whereby all that information comes from the DNA. That means what? If the mitochondria can produce an enzyme on its own, it means that it has DNA. And this DNA, we normally call it mitochondrial DNA. So we are going to see how genes in the cytoplasm are actually inherited from one parent to another. And we are going to see that the egg, which contains most of the cytoplasm, is the one which is actually, most of the genes which are found on the egg, the mitochondrial genes which are found in the egg are actually the ones which are passed to the next generation. For the sperms, you find that the sperm will lose most of the cytoplasm in the process of spermiogenesis. And in that case, if the mitochondria was there, it will disappear. So you find that those genes from the male parents in the cytoplasm are not inherited. So most of the cytoplasmic genes or genes found in the mitochondria, the ones which are passed to the next generation are the ones from the maternal side. That's why you hear people saying that when it comes to forensic, you check the mother, you can trace your origin by looking at the mother, the maternal cousins, everything maternal, because the mitochondria generation to another on the maternal side. like you guys, you have the same mitochondrial genes like your cousins, the maternal cousins, something like that. We are going to see that cytoplasmic genetic system, because these are now genetic systems which are found in the cytoplasm, not in the nuclear. Yeah, not in the nuclear. So I think that is the end. That is the end. Maybe we meet